All right, everyone. So in this lecture video, what I'm going to be going over is kind of a quick and dirty overview of what's happening with a muscle contraction. Okay, so the idea of a muscle contraction is where you have a skeletal muscle that's changing length, in this case, contracting, shortening, so that it pulls on the bone in order to get a particular type of motion. Okay, so with a muscle contraction, the big thing to think about is that it's utilizing an action potential. So an action potential is just where you've got some discrepancy in charges across a membrane that allows you to utilize that discrepancy to make something change. Right, so to either signal some other protein to do something else, or by changing concentration, you're changing the shape of something that's allowing the muscle to actually carry out its particular function, or in this case, the muscle to carry out a particular function. But you have action potentials across a lot of different places, like in nerve impulses, digestive things, all kinds of stuff that are going to carry out this particular thing. Okay. All right, so let's talk about the parts. So there are 5 parts to a muscle contraction as a whole. Okay, a lot of these muscle contraction parts deal with the movement of different ions across the membrane, specifically what's going on at the T-tubule and things like that. Okay, so let's start with our initiation. Okay, so let's talk about what it takes to get ready to get to a particular action. Okay, so let's start with the neuromuscular control. So what ends up happening is that you have this neuromuscular control center that's allowing you to send a signal. Okay, so what ends up happening is the signal sent at the neuromuscular junction, so the junction between where the nerve, right, from your central nervous system meets the sarcolemma. So remember, the sarcolemma is the cell membrane of a muscle cell. Okay, so we call that the neuromuscular junction, the point where those two things meet. Okay. Right, with our neuromuscular junction and the sarcolemma, what ends up happening is we end up sending a signal that releases calcium ions. So remember when we talked about the muscle cell, right? You have stored calcium ions within the sarcoplasmic reticulum that are then released. And so what ends up happening by releasing that signal, right, the nerve sends acetylcholine. Okay, so acetylcholine, or ACH, is actually the neurotransmitter that's responsible for muscle contractions. Okay, and what it ends up doing is this sending it across this gap, because your neuromuscular junction is kind of this little bit of space that's between the two, and I'll show you an image of that in your other resources that you can look at that allows for the muscle to actually send the signal. Okay, so by going and jumping that gap, acetylcholine causes the muscle to actually change shape. Okay, so what ends up happening is the acetylcholine ends up binding to the sarcolemma, and this ends up triggering that action potential I talked about before. So it actually encourages the cell membrane to move different ions across it, right, in order to send the signal down the muscle fiber to cause these sarcomeres to start contracting. Okay. So just a quick thing, neuromuscular control, right? You're sending a signal at the neuromuscular junction, so something from the brain sends it down to the junction. The junction between that encourages the release of calcium ions, which sends from the nerve to the muscle acetylcholine. Acetylcholine then jumps the gap and triggers on the sarcolemma, so the cell membrane, and we get the action potential. Okay, let's talk about the muscle action potential. Okay, so there's a lot of things going on here, right, where the acetylcholine is binding to a particular receptor. So when it binds to that receptor, it ends up releasing a protein, so opening up this channel that releases sodium ions. Okay, so we know that ions, because they're charged, don't readily move across a cell membrane. Okay, by changing the shape of a particular channel, allowing it to open, we're going to have an ability for these ions to move across the membrane. Okay, so we're going to end up going from an area where there's a high concentration to a low concentration. So in this case, when we're removing our sodium ions, we're decreasing the resting potential of what's going on. Okay, so the sodium ions that are outside of the cell actually enter into the sarcolemma, and that causes it to depolarize. You no longer have an end side that's positive and a side that's negative. Both sides end up having the same charge. Okay, so what ends up happening is when we depolarize from the positive end and we change it, that depolarization ends up getting spread down the membrane. Okay, so the sarcolemma ends up changing from being this positive to negatively charged thing to where there's no longer a charge on either side. Okay, because that happens, right, your membranes don't like to not be polarized, essentially. It's kind of like the summary of it all, is that in order to balance out the influx of calcium or sodium ions, potassium channels open up in regions where it starts to depolarize to allow us to repolarize those areas. Okay, so we end up sending potassium ions in one direction as sodium ions start to enter. Okay, so those begin to leave, which again rebalances the charges. So again, we have one side that's more positive than the other. Okay. right? So after that potential spreads down, right, and we get our kind of changing back to where one end is positive, one end is negative, those channels end up changing shape. So we end up closing the channels, which doesn't allow for sodium ions to move anymore, right? So by taking something that's positive to negative one end, adding the sodium ions into the cell, which depolarizes it, potassium comes in, repolarizes it, which means sodium can't move anymore. Okay, so what ends up happening by opening those channels, we repolarize it, and then that allows the potassium to then get back and reabsorbed, right, back to where we need to go in order to reset the signal. Again, this action potential happens so fast, like nanoseconds fast, right? We get this very rapid shift of sodium ions and potassium ions back and forth, right, that gets reset over and over and over again very quickly. Okay. The problem with this action potential is it can't go on forever, right? Your muscles get fatigued. They don't move quite as fast. So we have a harder time exchanging these as the muscle gets fatigued. So just that's one thing that happens with muscle fatigue. Okay. So now that we have this action potential, let's talk about what actually happens to the muscle. Okay. So as we have this action potential running down the membrane, okay, the action potential travels down these T-tubules. So remember, T-tubules are embedded within the cell membrane. They actually go down into the actual sarcomere, or sorry, they go down from the sarcolemma down into near the sarcomeres, right? And what this ends up doing by sending out these calcium ions in the sarcoplasm, right, again to that space outside, the calcium ions are now available for the sarcomeres to utilize. And by binding to the calcium, this is actually what makes the particular parts of your muscle fiber start to contract and actually change shape. So what ends up happening is the calcium binds to the troponin, which is one of the muscles that are, or one of the fibers that's part of the sarcomere, right? And what that essentially makes it available for by attaching the troponin is actually makes actin available for and myosin available to actually interact, okay? So by having that troponin, that kind of block, right? This is molecule that's there that's preventing the muscles from actually contracting. By putting calcium ions on it, you actually change its shape, opens it up, allows for actin to come in, right, to get on that. And what happens is actin and myosin can now interact with each other, okay? So by doing that, right, it allows us to actually then have the muscle fiber start to contract, okay? So just to summarize everything that happened, neuromuscular control has come in, the signal is sent to the neuromuscular junction, right? Calcium ions are released, okay, which sends a signal for acetylcholine to be released across the gap, right? Acetylcholine binds to the sarcolemma, which causes the action potential, okay? We have a sudden influx or change of sodium and potassium ions. So sodium comes in, potassium comes out, and polarizes it. By polarizing it, we get more calcium ions released not into outside the cell, but into the sarcoplasm. And then that calcium ions can bind to the active sites of your sarcomeres, which allows them to now be able to contract. Okay. So let's look at what actually happens with with our contraction. Okay, what's going down there. Okay. So with muscle contraction, our next step is we're going into what's called our sliding filament theory of contraction. Okay, so the sliding filament theory, right, is pretty well understood. The idea that you've got these different molecules that are running across each other. Again, there's some things about it that aren't quite completely understood. So if that's something you want to pursue and look at later on, you can. But again, looking at the specific details that are happening, because like I said, this is an extremely complicated process. But what's happening here is that during the muscle contraction of the thin and thick filaments, so actin and myosin, they essentially slide past each other, right? So they slide and kind of ratchet next to each other and allows it to start to contract together. Okay, so what ends up happening as we move through this, right, myosin, the thick filament, right, is actually going to have a part of it that attaches to the actin. Okay, and that requires ATP to happen. So what's happening is that myosin, like the head of it, pivots and actually pulls on the actin. Okay, and pulls it towards the center. So you're taking that thin filament and essentially like ratcheting it over, right, and then that resets itself over and over again. It starts ratcheting it down. Okay, so the head detaches where ATP binds, and then myosin resets and rebinds. So just like is ratcheting down the line. Okay, and it repeats this step over and over and over again until we get the actin filaments basically all the way touching each other almost, where the H band inside of our sarcomere disappears. Okay, so again, if we looked at like a muscle that's under full contraction under a microscope, it's going to be like really dark because again, there's not a lot of space because everything is so wound together. Okay, so what's ended up happening here with our sarcomere, if we're just talking about structural relationships that are happening here, is that our sarcomere shortens, right? Our Z lines become closer together. So essentially what we see is our H band gets gone, it disappears. Our A band gets a lot darker, and our I band, so the difference between our Z line and Z line, or sorry, from the end of one Z line to the other one, is going to shorten, right? So the fiber itself overall gets shorter as a whole. Okay, so again, what's happening here is that myosin is just latching on actin and just pulling it over, right? As you can see with my like really awesome example with my hand waving under here. But essentially we're just like ratcheting and pulling it, right? So we're just like, if you're thinking about like climbing a rope, it's like reaching that next knot, right? And then pulling that and then grabbing the next knot and pulling that so that you can bring those muscle fibers closer together. Okay? So the last thing that we do when we're done, right? So say the muscle has fully reached its contraction point, is that the muscle is ready to reset. So we've repolarized everything, right? And that ends up sending the signal for the calcium ions to return back to from the sarcoplasm and get stored into the sarcoplasmic reticulum. And then troponin, which is again that muscle that block the active sites on your actin and myosin. So troponin on your actin, tropomyosin on the myosin, right, ends up coming back, right? It's the calcium's no longer there. Everything changes back to its original shape. Allows troponin to go back in, and it resets. So it just covers it up. So the muscles like have to release, right, and then they relax. Okay, so like with any of the stuff that's going on here, it's all about changing the charges of the situation, and what ends up doing there is just changing the shape of the muscle fibers, right? Remember when we talked about enzymes and active sites, kind of the same thing that's happening here, right? The idea that you're changing the charges, which is changing the shape of the particular molecule, which is causing it to do what it's doing. Okay, so all this stuff will start to reset. So with our muscle contraction, the thing that I'm really interested in you guys, or want you guys to know, is like, what are the main steps that are happening here? What are the big pieces that come together in order for your muscles to contract, in order to get it to do what it's doing? Okay, so what you're seeing. Okay, so that's it for muscle contraction. Okay, I'll have some additional resources for you, some videos you can watch to describe what's going on in a little bit more detail. Again, diagrams with models and things like that you can use to help you understand what's going on with muscle contraction. Okay, all right, so if you got more questions, let me know.